Hello everyone, today I will be discussing about different models of bacterial growth kinetics. Microbial growth is measured by an increase in the cell's macromolecular and chemical constitution, and each microbe's growth pattern is unique. Microbes' cell growth and division are inextricably linked as bacteria divide via binary fission, yeast cells via budding and the viruses via intracellular division. Because nutrient availability influences mammalian cell growth, a threshold cell size is required for DNA synthesis and mitosis. As a result, each class of microorganism has a distinct growth pattern determined by their cell cycle and cell division. As a result, each class of microorganism has a distinct growth pattern determined by their cell cycle and cell division. Understanding the growth kinetics of various classes of organism serve as the foundation for the fermentation process in order to achieve optimal product concentration. Microbial growth kinetics, or the relationship between a microbial population specific growth rate and the substrate concentration, that is S, is an indispensable tool in all fields of microbiology, including physiology, genetics, ecology and biotechnology, and is thus an important part of basic microbiology education. As we all know, bacteria are single celled microorganisms that reproduce through a process called binary fission, where one bacterial cell divides into two identical daughter cells. You can see in pictorial representation, bacterial growth refers to the increase in the number of bacterial cells in a population over time. Bacterial growth kinetics refer to the study of how bacterial populations grow and reproduce over time. Various mathematical models have been developed to describe the different phases of bacterial growth, taking into account factors such as population size, time and environmental conditions. Bacterial growth kinetics experiments require specific conditions, equipment and methodologies to accurately study how bacterial populations change over time. Bacterial growth kinetics, which describe how bacterial population change over time, are influenced by a wide range of factors. These factors can either promote or inhibit bacterial growth, and understanding them is essential for various applications in microbiology, biotechnology and other fields. Let us look into some of the commonly used models for bacterial growth kinetics. First one is exponential growth model, second is logistic growth model, then Gompertz growth model, Barani model and Monod model. So, we will take first the exponential growth model. It is also known as doubling time model. This is one of the simplest and most commonly used model for bacterial growth. It assumes that bacteria grow exponentially, meaning that the population size doubles at a constant rate over a fixed period. The mathematical equation for exponential growth is Nt is equal to N0 into 2 raised on T upon generation time, where N is population size at time T, N0 is the initial population size and T is the time in hours. Generation time is the time it takes for the population to double. Exponential growth occurs when resources are abundant and limiting factors are absent. In reality, such conditions are rarely sustained for extended period. The rate of population growth, the R, is constant as long as environmental conditions remain ideal. Bacteria in the exponential growth phase double in number with each generation. The population size increases rapidly during the exponential phase, leading to a step curve on a population growth graph. You can see in the pictorial representation. In nature, exponential growth is unsustainable over the long term because resources become limited, waste accumulates and other factors, for example, competition, predation and disease eventually slow or halt growth. The exponential growth model is a simplification and serve as a theoretical reference point for understanding how population can grow under ideal conditions. In practice, bacterial growth often follows more complex patterns, including a transition to the stationary and death phase as resources become depleted. Next one is logistic growth model. The logistic growth model takes into account environmental factors and population density. It assumes that bacterial growth starts exponentially but levels off as resources become limited. The equation for logistic growth is Nt is equal to k upon 1 plus k minus n0 upon n0 into e raised to minus r into t, where Nt is the population size at time t and 0 is the initial population size and k is the carrying capacity. Maximum population size is environment can support. R is intrinsic growth rate and t is the time. The logistic growth model is a more realistic representation of population growth compared to the idealized exponential growth model because it considers the constraints imposed by limited resources and environmental factors. The logistic growth model is widely used in ecology to model the growth of populations of organisms in natural ecosystems as it better reflects the real world dynamics of population under resource constraints. Next model is the Gompertz growth model. In this model, it is often used for describing bacterial growth when there is a lag phase before exponential growth and when growth slows down as resources are depleted. The equation is as follows: Nt is equal to N0 into e raised to minus e raised to mu max into e upon N0 into t minus t lag, where Nt is the population size at time t and 0 is the initial population size, mu max is the maximum specific growth rate, t lag is the duration of lag phase and here t is the time. The Gompertz model is particularly useful for describing situations where population experience slowing growth rates over time, potentially due to resource limitations, competitions or other factors. Next one is the Baranyi model. The Baranyi model is used to describe the growth of bacteria in non-isothermal conditions and incorporates lag phase, exponential growth and stationary phase into its equations. It is often used in predictive microbiology. In Barani model is versatile and can be used to predict microbial growth under various conditions, including changing environmental factors. It is often used in food safety and microbiology research to assess the growth of pathogenic and spoilage microorganism in food products. These models provide a framework for understanding how bacterial populations change over time and are valuable tools in fields such as microbiology, food safety and biotechnology. Researchers use experimental data to fit these models and make predictions about bacterial growth under different conditions, helping to ensure food safety, optimize fermentation processes and study microbial ecology among other applications. Next one is the very common model, that is monode model. The monode model, also known as the monode equation, it is a mathematical equation used to describe the growth rate of microorganism, particularly bacteria, as a function of the concentration of a limiting nutrient in their environment. It is named after the French microbiologist Jacques Monod, who developed this model in the mid 20th century. The monode model is widely used in microbiology and microbial ecology to understand and predict the growth of microorganisms under specific conditions. The monode equation is typically written as mu is equal to mu max into S upon Ks plus S, where mu represents the specific growth rate of the microorganism, that is how fast it is growing under specific conditions. Mu max is the maximum specific growth rate, which is the growth rate when the concentration of the limiting nutrient S is abundant and not limiting growth. S is the concentration of the limiting substrate or nutrient that the microorganism needs to grow. This could be, for example, the concentration of a carbon source, nitrogen source or any other essential nutrient. Ks is the monode constant, which represents the concentration of the limiting substrate at which the microorganism's growth rate is half of its maximum, that is mu max upon 2. The monode equation describes how the specific growth rate of a microorganism increases with increasing substrate concentration until it approaches its maximum growth rate, that is mu max, as the substrate concentration becomes abundant. However, as the substrate concentration decreases below the monode constant, that is Ks, the growth rate decreases significantly, indicating that the microorganism is limited by the availability of the substrate. The monode model is particularly valuable in environmental microbiology, wastewater treatment and industrial fermentation processes, and it helps researchers and engineers optimize conditions for microbial growth and metabolism by understanding how nutrient concentrations impact growth rates. Applications in bioprocess optimization, in biotechnology and industrial microbiology understanding bacterial growth kinetics is crucial for optimizing bioprocesses such as the production of biofuels, enzyme, pharmaceuticals and bioplastics. Researchers can use growth models to determine the ideal conditions, for example, temperature, pH, nutrient concentration for maximizing microbial biomass or product yields. In case of fermentation, bacterial growth kinetics are applied to fermentation processes used in brewing, winemaking, cheese production and the production of various fermented food. These models help to control fermentation parameters and ensure product quality. In food preservation, in food microbiology, predicting bacterial growth kinetics help ensure the safety and shelf life of food products. Researchers and food manufacturers can use these models to determine the appropriate conditions for food storage and preservation, including temperature, humidity and packaging methods. In case of medical microbiology, bacterial growth kinetics are essential for understanding the pathogenesis of infectious diseases. Studying the growth of pathogenic bacteria helps in developing diagnostic tests, determining antibiotic susceptibility and designing treatment strategies. In antibiotic development, understanding bacterial growth kinetics is crucial in the development of antibiotics and antimicrobial agents. Researchers can use models to study how bacteria respond to various drugs and to design more effective treatments. In environmental microbiology, bacterial growth kinetics are applied to study microbial communities in natural environments such as soil, water and sediments. These models help researchers understand ecosystem dynamics, nutrient cycling and the impact of environmental changes on microbial populations. In wastewater treatment, bacterial growth kinetics play a role in designing and optimizing wastewater treatment processes. Microbes are used to break down organic pollutants and growth models help determine the efficiency of treatment systems. In case of bioremediation, in environmental science, growth kinetics models are used in bioremediation efforts to clean up contaminated sites. Certain bacteria can degrade or transform pollutants and growth kinetics help assess the rate at which this occurs. In microbial ecology, researchers use bacterial growth kinetics to study the dynamics of microbial communities in different ecosystems, helping to understand how various factors including temperature, nutrient availability and competition affect community composition and function. In research and education, bacterial growth kinetics models are fundamental in microbiological research and are often taught in microbiology and biotechnology courses to help students understand how bacteria multiply and respond to different conditions. While bacterial growth kinetic is a valuable tool in microbiology and related fields, it also has certain limitations and disadvantages. Here are some of the disadvantages associated with the study and application of bacterial growth kinetics. First is simplified models. Bacterial growth kinetics models often assume idealized conditions such as unlimited nutrients, a constant environment. In reality, microbial growth in natural setting is much more complex with fluctuating conditions and interactions with other microorganisms. Variability. Bacterial growth rates can vary widely among different species and strains. Applying a single growth model to diverse bacterial populations may not accurately represent their growth behavior. Lack of consideration for lag phase. Some growth models skip the lag phase, which is the initial period of slow or no growth when bacteria are adapting to a new environment. Ignoring the lag phase can lead to inaccurate predictions, especially in practical applications like food safety and bioprocessing. Environmental factors. In environmental factors, bacterial growth kinetics models may not account for all relevant environmental factors such as pH, temperature, oxygen level and the presence of inhibitory substances. These factors can significantly affect growth rates, but may be oversimplified in some models. Then complexity of microbial communities. In natural environments, bacteria often exist as part of complex microbial communities. Traditional growth kinetics model typically focus on individual species and do not consider interspecies interactions and competition. In genetic variability, bacterial populations can exhibit genetic variability leading to differences in growth rates and adaptation to changing conditions. Some models may not capture these variations effectively. Non-exponential growth. While exponential growth is a useful concept, many bacterial populations do not exhibit continuous exponential growth for extended periods. Growth rates can change due to nutrient limitations, environmental fluctuations and other factors. Applicability to specific cases. Growth kinetics models developed for one bacterial species or strain may not be directly applicable to others. Each bacterial population may have unique growth characteristics. Labor intensive data collection. Accurate determination of growth parameters often requires frequent sampling and labor intensive measurement such as cell counting to turbidity reading, which can be time consuming and resource intensive. Limited predictive accuracy. Growth kinetics models provide valuable insight into bacterial growth patterns, but their predictive accuracy can be limited when applied to complex, dynamic and heterogeneous systems. Despite these disadvantages, bacterial growth kinetics remains a fundamental tool for understanding and modeling bacterial growth under controlled laboratory conditions. Researchers and scientists often use this model as a starting point to study microbial populations, but they must aware of the limitations and consider real world complexities and applying the result to practical situations. Now, I will talk about research and advancement in the field of bacterial growth kinetics. I will also discuss the futuristic research in this field. Population heterogeneity. In single cell model, imagine a big group of friends walking together. Each friend might walk at a slightly different speed and some might even take short break while others keep walking. In the same way, bacterial cells in a population do not all behave the same. They can grow at different rates and some might pause their growth for a while. Single cell models are like special cameras that focus on one friend at a time. Instead of looking at the whole group, they watch each bacterial cell separately. These models help us understand how some cells grow faster or slower than others and why some might stop growing for a bit. In subpopulation dynamics, discuss how models can capture the growth patterns of different bacterial subpopulations within a mixed culture. Now, let us say your group of friends is not all the same age, some are kids, some are teenagers and some are adults. They might not all walk at the same speed, right? Bacterial population can be like that too, with different groups of subpopulations growing in different ways. In evolutionary dynamics, mutation and selection models explain how bacterial growth models can be used to study the evolution of antibiotic resistant and their traits. Imagine you have a collection of colorful marbles and you want to find the rarest, most unique one. To do this, you keep making small changes to the marbles, like painting them slightly different colors. After each change, you pick the most unique marbles and make more changes to it. Over time, you end up with a super rare, one of a kind marble. Bacterial growth models work a bit like this marble game, but with the bacteria. Bacteria can change or mutate their traits just like the marbles change colors. Some of these changes might help them survive when they face something harmful like antibiotics. The models help us understand how these changes happen and which bacteria traits are more likely to survive and spread, like finding the rarest marble. In cooperation and competition, models that address bacterial interactions such as cooperation and competition within population. Imagine you are playing with a team sports like soccer. In your team, some players are really good at scoring goals while others are excellent at stopping the opposing team. To win, your team needs both type of players to work together, but sometimes a player may want to score all the goals but not to cooperate others. Then data driven approaches, machine learning and deep learning. How machine learning techniques can be applied to predict bacterial growth pattern using large databases. Imagine you have a super smart robot friend. This robot can learn from watching you and your friends play different games. Watches your play soccer, chess and wide and hide and seek. After watching a lot, the robot becomes really good at predicting who will win and how the game will go. Even it is a game it has even seen before. Then big data integration. Use of dynamics data, genomics, transcriptomics, proteomics to refine bacterial growth models. Imagine you have a big puzzle with lots of pieces. Each piece has a tiny picture on it. To solve this puzzle, you need to put all these pieces together in the right way. Now think of the pieces as information about bacteria. Some pieces are about the bacteria genes or genomics, some are about what they are doing, transcriptomics, and some about their protein, proteomics. Big data integration is like solving this puzzle. Scientists gather all these pieces of information from bacteria and put them together. Now the emerging techniques, microfluidics and lab on a chip. devices, how these technologies enable real time monitoring of bacterial growth and its integration with modeling. Imagine you have a tiny laboratory and on a computer chip just like a small piece of your computer. This mini lab can do amazing things. It can mix tiny amount of liquid like a scientist mixing ingredients for a recipe, but instead of baking cookies, it is a studying bacteria. Microfluidics and lab on a chip devices like these mini labs, they let scientists control tiny amount of liquids where bacteria live. This helps them watch bacteria grow in real time. Then single cell imaging, the potential of advanced microscopy techniques for collecting data to validate growth models. Imagine you have a super duper microscope that can see things at the scientist level. With this microscope, you can watch individual bacteria at your backyard through a magnifying glass. You can see what each bacterium is doing and how it is growing. Single cell imaging like using this super duper microscope. Scientists use advanced microscope to zoom it or individual bacteria and take pictures on videos of them. Thank you.